Yes friends, I am Professor Kumar and you are learning at Galaxy Online Learning, a venture of Galaxy Tutorials. Today we are doing a very important chapter, Human Health and Disease. Earlier it was a very small chapter, just for 1 mark or 2 mark. Now its weightage in theory and NEET has increased considerably because lot of sub-chapters have been brought into this chapter and has become a major chapter, Human Health and Diseases. Okay, first, what is health? Health is a state of complete physical, mental and social well-being, body, mind and society. Only you don't have disease or your body is looking perfect will not do. WHO definition. This is a conceptual definition. How do we see in everyday life? Everyday life, operational definition. Body should operate. Condition or quality of the human organisms showing adequate function, more than enough functioning to live a normal life. We don't want you to be a superhero. In given condition, wherever you are staying, whatever is your genetic composition and how is your environment. Please remember, health simply does not mean absence of disease or unfit, I have muscles and all that. WHO, I remind you once again for NEET purpose, physical, mental, social well-being. Health is basic right, birthright of yours, okay, included in your basic rights. It shows metabolic and functional efficiency, your digestion, your physiology, all working carefully. And the rules we make to keep good health is called hygiene. Have a look at what is a current concept of what is your well, 360 degrees. Environment, where you live, emotions, mental, finance, money is the root of all problems, social, society, spiritual, mind, occupational, work and satisfaction and earning, physical, your strength of the body, intellectual, your mind. Now you can see all-round development is good health. What we should do, this is what we study from lower classes. First, balanced diet you should eat, you should wash your hands and all that. Hygiene, clean drinking water, community hygiene, personal hygiene, physical exercise, at least 20 minutes a day. And we should have knowledge how diseases are passed on so that we can prevent them. We should dispose of waste properly so that those flies and mosquitoes and worms and bacteria are taken care of, not dump them in just water or soil. What makes good health? Based on that 360-degree approach, we see income, money is again most important, social support, how are your friends and support, employment, where you work, you spend almost more than 3/4 of a day, physical environment, your surroundings, education, as a child, how healthy you were brought up, your biology of the body, your genes, health services available so that small problems can be solved, and how you keep personal health practices so that you are keeping it more fit. Immune system, we are going to see in details now. First part, what is an immune system? System which protects us from various infectious agents. Number 2, what is a resistance? Ability to prevent damage or disease through defense mechanism. Please remember, now since there is COVID everywhere, everybody is not getting it because immunity is present. Immunity is old definition, resistance towards injury by pathogens and their products. Okay, from time immemorial we always define immunity like this. It is a freedom or exemption from the disease and pathogen. What are the modern definitions? Immunology for your NEET and MCQs. Immunology is the study of immune system. Immune system is cellular and molecular components. Remember now, your WBC types and chemicals made by them are responsible for immunity. Immunity is old definition, protection from infection. New definitions, we will add even non-infectious foreign substance to it. Any chemical which enters like pollen or some food item and you get allergy is also now included in this. So collective, total, coordinated, step-by-step response to these foreign objects by the body is immune response. So now we define immunology is study of cellular and molecular mechanism which allow the body to first not destroy itself, destroy the foreign. Second, whatever is foreign not of the body be destroyed. Edward Jenner is the father of immunity. He first made cowpox vaccine, which we'll see in the next slide. Immunology is now again defined branch of biology, etc. Now, protection against disease is only one part of immune response. Now, the reaction of the body to any chemical, organ, tissue put into the body also comes under immunity. Just to remind you, now immunity definition is broader. Let's see what Edward Jenner did. So, this is historical. Sarah Nims, a milkmaid, first got cowpox. She was milking the cow from the udders of the cow. Now James Phipps is inoculated by, he took a needle and took the pus and gave it to him. James gets mild smallpox, cowpox. Then you collect the scabs and give it to intermediate. So this is how we are diluting this and causing more and more precise response. Again the scab is given back to James. Now he does not get any infection. He does not show any symptoms. Now protection is completely, he is immune to the cowpox germs. Please remember, self and non-self distinguishing is very important. Otherwise your body will get destroyed. Now any foreign substance which does not belong to you is called an antigen because it will produce a reaction. And the reaction produced by the body in terms of protein and chemical is called antibodies. So again we remind you, you have got certain markers, molecules on top of your cell like RBC or cell, which your WBC is happy with and will not attack. As soon as foreign markers come, the antigen will recognize it as a foe, enemy. For example, bacteria have it on the top, virus have these spike-like things. And here you can see a pathogen has all these molecules on the cell wall, etc., cell membrane. So the pathogen attacks the cell, cell recognizes the antigen and produces antibody. What are the types of immunity? Just one more question. Innate, you're born with, we are going to see in detail, this is just a chart. Adaptive, which you acquire. Innate will be anatomical barrier, physiological barrier, phagocytic barrier, inflammatory barrier. Okay, anatomical skin, how your body is constructed, tight so that germs don't enter. Physiological barrier we have like tears and secretions. Phagocytic barrier we have plain WBCs. Inflammatory barriers, skin gets swollen so that bacteria don't enter. Now adaptive is active or passive. We put the weakened germ or we put the antibody directly. So active also natural immunity and artificial immunity. Okay, and passive also natural immunity and artificial immunity. We are going to see it in details. First we'll go for that innate immunity. Inborn, you get by birth. That is because of your genes and DNA. Third, it's your defense system of your body. Fourth, you have never met a pathogen or chemical before. It is non-specific, will fight with everything. And it has all these barriers which prevent a newborn baby survives and meets millions of germs on birth is because of innate immunity. Have a look at the diagram. The tissue, epithelial cells are tightly packed. We have got phagocytes, we have got dendritic cells, we have got natural killer cells, NK cells, and we have got chemicals like complement which go and destroy. We are born with this. Now, in detail, epithelium. How does the epithelium? Skin when intact. If it's broken, germs will enter. Mucus covers most part of your body, nose, esophagus, etc. And sweat, which has a lot of salt. And sebum, sebaceous gland of your skin. Respiratory tract mucosa, hair in the nose. Okay, mucous lining of the epithelium. Cilia. These keep pushing all the particulate matter and pathogens into the mucus and we cough it out. Cough reflex is important. We should not suppress cough. We should cover our mouth when we are coughing. Okay? We have saliva. We have HCl in the stomach, gastric glands making it. We have urine which throws out. We have spermine and zinc in semen. Conjunctiva is protected because of lacrimal tear secretions. When you are sitting on a computer or you are reading the book whole day and you don't blink, your eye becomes dry, lacrimal secretion or tears are absent. Now they contain lysozyme, discovered by Alexander Fleming. These are thermolabile, low molecular weight, basic protein, but destroy a component of the cell wall of the gram-positive bacteria. That's why when other bacteria, gram-negative or virus comes in, it cannot protect. We get conjunctivitis, red eye or pink eye. Okay, but most cases it protects. Just have a look at the chemical barrier. Sweat, oil, saliva, tear, mucus, general. Okay, we will not put complement, etc. We will be seeing in details. What are some physical and chemical? Name some. Skin, mucous membrane. Cilia, mucus, reflex, coughing like. pH, lysozyme, fatty acid, defensin molecules. Normal bacteria in the body protects you. Genetic resistance, different in different species. Individual differences, two people also are different. So we don't get diseases or pathogens from other animal or plants because of genetic resistance. Blood contains antimicrobial substance called complement system. More than 30 serum proteins are there. They are inactive. Whenever the pathogen comes, it is activated and a waterfall, complement cascade, one trips the other, domino effect takes place. Okay, and the pathogen is destroyed. The cells, when viruses come in, make a warning protein called interferon. They are cytokines, soluble proteins. Now these go to every cell of the healthy cell of the body, the neighbors, and tell them, get prepared, tell the WBC, start fight, virus has come in. Just have a look like a cartoon. Cellular factors. Phagocytic cells. They are natural defense cells of the body. They capture the foreign particle or pathogen and destroy it. They were discovered by Metchnikoff 1882. All the neat extras are here. Who call them microphage and macrophage. Today we have full study. Natural killer cells are present for tumors, cancer and viruses. You have a look at just like a Amoeba diagram it is, WBC through the pseudopodia, engulfs it, makes it like a food vacuole, secretes enzymes and destroys the pathogen. Natural killer cell, 2 marks. It's a type of WBC. It can recognize self and non-self. They can destroy cancer cells. People who have less natural killer cells because they have inherited immune disorder are more prone to certain viruses. They get many viral diseases. These are present in the blood and they can move by pseudopodia, pedaces into various parts of the body. Fever. When you increase body temperature, more the temperature, faster is the reaction to destroy. So, and interferon is produced and you recover from minor viral infections like cold, cough, etc. Now, when the local response is not working, the whole body will respond. The activated macrophages release a chemical interleukin-1, which travels the blood, goes to hypothalamus, and the body readjusts and temperature increases. 103, 104. Why? Higher temperature stops the growth, increases phagocytosis, increases repair, liver and spleen will store iron and blood iron will come down so that other proteins now can act faster. Bacteria need large amounts of iron to grow. So shut off the iron supply. Acute phase proteins. Whenever we have injury, we release plasma proteins called acute phase proteins. They enhance our resistance. They prevent further tissue damage. They promote inflammatory lesion, swelling local. These are C-reactive protein, mannose binding protein, alpha-1 acid glycoprotein, serum amyloid protein, etc. Just for info and your NEET exams. Now you see acute phase. Now you have seriously injured. 50 to 1,000 times these proteins will increase. Example, CRP made in the liver, normally it is less than 1. C stands for carbohydrate, for clarification. And when we take a blood test and CRP is very, very high, we know the body has got internal injury if external cannot be seen. So let us have a look at the cells which take part in the innate immunity. First, basophils and mast cells, they release chemicals causing inflammation. Neutrophils are the ones which capture and destroy. Eosinophils, particularly antibody, those which are marked, are destroyed. Monocyte and macrophages, antigens, chemicals. Dendritic cells, or everything, and they are activating other cells. They are like a monitor. NK cells, cancer and virus. So these are the cells, and this is what you will tick in the exam for MCQ. So first line of defense, pathogen has come, virus has come, bacteria comes, skin, ciliated cell, lysozyme, coughing, vomiting. Nothing happens, you are not well, they are growing. Second line, blood clot, then histamine is released, redness, scratching, swelling, cytokines are released, and leukocytes come into action to fight. Acquired immunity, you get it. It can be adaptive or specific. It involves forming antibodies, which will then go and fight with the antigens. 4 things are seen in this. Diversity, different antigens, so different types. Specific, each and every antigen has its antibody. Memory, we keep record who we met earlier. Okay, so second time we are more ready. And lastly, we will not destroy our own cells. So have a look. Specific, memory, self, non-self, and 2 types of cells are involved, T and B cell, for making the protein and for doing the reactions. What are unique features of these memory? Again, specific, diverse, self, non-self, and memory. First encounter produces memory. Second, stronger and quicker response. So 2 marks, 4 points required about adaptive immunity. Now, acquired immunity can be active passive for natural and active passive for adult. What are these? First, you recover from a disease, you get naturally immune for next time. Mother to fetus, she gives to a newborn child through placenta. These are natural. Artificial, I give you vaccine, weak germs, and you produce antibodies. And passive, I give you directly antibodies because you can't make the antibodies using anti-serum. Now we are doing plasma therapy in COVID, which is passive artificial acquired immunity. So, what to write in the notes? Active, when you have met with the antigen, encounter is already there. Or we put little in your body, so we stimulate your cells. It may be natural, it is after meeting the pathogen, it is long lasting, okay, throughout normally for life, and you never get again measles, for example, or you are vaccinated and all. Artificial, as I told you, polio, BCG we give you at birth so that you don't suffer from them later. Passive. Passive you get from the mother, colostrum, first milk, breastfeeding, okay, short-lived because you don't feed mother's milk throughout the life. Artificial, you have to prepare it in a horse or for example other animals, get the antibody out and then inject. For example, for rabies we use hyper-immunized horses, species specific, so they don't suffer much and we get the antibody for it. Just as a lighter note, what the cells do, so you can just have a look, so that it's fixed in your brains. You don't have to draw anything like this or write in the exam. Yeah. What is the role of lymphocytes? They are the main cells of immune response. They come from stem cells, hematoblast, in the liver and bone marrow. They are B cells. They differentiate in the gut, in bursal lymphoid tissue, tonsil, pairs, patches in the intestine. They are called bursal or B cell. They produce antibodies, humoral antibody mediated immune system, AMIS. So have a look. So we have this B cell and antibodies are attached to the B cells. When bacteria come, they get fitted on them and they are recognized like this. So the cell gets activated now. The other cell is T cell. It is made in the thymus gland and it's called T cell mediated immune response. CMIS, cell mediated immune response. They are traveling throughout your body fluid, blood and lymph and circulating in the body. What are the types of T cells? T cells are of 4 types. First, helper. They produce lymphokines or chemical messengers which will warn other T cell, will tell the B cell also and the macrophages. Second, killer T cells or cytotoxic T cells. They are suicide cells. They go attack and kill themselves and the pathogen or the invading cell. They secrete perforin, make holes so that the pathogen leaks. Suppressor. The suppressor tells us, like monitor, don't make noise, so our body does not fight against its own cell. And memory cells, they keep a memory of the encounter next time the pathogen is seen, immediate production, immediate response, and faster and higher response. You can have a look, diagrammatic, type of lymphocytes, T, B, and NK cells. Okay, just for understanding. We are supplementing our ideas, making them stronger in your brain by bringing it more than one time. Let us write a short note. Lymphocytes are T lymphocyte, B lymphocyte are natural killer cells. Second, they can migrate and travel and they can engulf organisms or antigens. They are of 2 types, T cells which mature in thymus and B cells which mature in bone marrow. So summary or concise brought here. How do the B cells work? First, as I told you, the antigen fits into the receptors, then the T cell will give the information, B cell gets activated, he makes Xerox copies very fast, so clone of plasma cells and memory cells are formed. Now they make glycoproteins called antibodies which are circulating in the blood. The antibody molecule binds to the membrane of the invading cell and that is how they are marked or destroyed later. Have a look. Okay. First, antigen meets macrophage, WBC, general soldier of your body, activates helper T cell. This, when antigen comes to it, activate the B cell makes copies, clones, memory cells, plasma cells, then plasma cells make antibodies, memory cells keep giving the blueprint and ready for the next action, next time. How do the T cells work? Helper T cell tells the B cell tells the plasma cell secretes antibody. Only a part of the earlier slide showing you the role of T helper cells. How are B cells activated? Virus invades the cell, B cell becomes lymphoblast, lymphoblast becomes plasma cell, lymphoblast makes memory B cell, plasma cell makes antibodies. As I told you, we are supplementing our information. Now, antibody has 3 main points. First, agglutination. It can gather, okay, the invaders and pathogens into a mass so that WBC can chew it up. Opsonization. They mark, like how your t-shirt has number 10, number 8, okay, so they mark so that WBC knows which cell to capture. Third, they neutralize the chemicals, toxin, like tetanus, you take tetanus serum. Okay, have a look. Okay, how the action was diagrammatic so that you can, so you see here, opsonization, neutralization, agglutination, complement making holes through those proteins, precipitation like alum chemical, it will bring group total down because the mass now increases. Antigen presenting cells, they first capture, then they make the signal on top of their body molecules, and third, they travel and tell everybody, helper T cells get activated. They present the antigen by breaking it into pieces and putting it on their outside. Okay? You can see how antigen is captured by various antibodies. So bigger masses, and of course others will also get attached. Big masses form, so precipitate, etc. is easier. What are antibodies? Glycoprotein, sugar plus protein. Specific. Third, they are called immunoglobulins. Fourth, produced only when antigen is present. Made by plasma cells, which are a part of B lymphocytes. 2000 molecule per second they are made. See the rate. How do antibodies look? Y shape, 4 chains, polypeptide, too heavy, too light, and together joined by disulfide, sulfur-sulfur bonds. And in the middle, where the stem begins, they have got a hinge region, so that a little mobility is there. On the top is variable region, at the bottom and middle is the constant region. The variable region changes depending upon what antigen it has to capture. For example, you have a lampshade. The bulb holder is the variable region. You can change the holder for the type of bulb, but the lampshade does not change. That is a constant region. They are mostly bivalent. Why? So 2 antigens can be caught at 2 different sites. Have a look. Diagram. You have to label the diagram, draw the diagram. Comes in the board exam. Of course, you don't have to color, make boxes. You can draw straight lines. But this is how it looks. What I taught you. So 2 marks. Also have a look more in details. Okay. C terminal for the carbon and N terminal for the nitrogen. That's how we said top and bottom. And remaining is same. So everything is labeled again to show you the differences. Diagram is same, but for labelings we have made 2 diagrams. Okay. See how antigen binds in this one heavy chain, one light chain, and at the variable region, okay, it binds. What is binding is epitope, where it is binding is paratope. They are opposite, complementary shape to each other. Then only, that's why it is specific binding. Remember what is epitope, what is paratope, and study of antigen-antibody reaction is serology 1, 2, and 3. And paratopes are present only on the variable, not on the constant region. And what you call antigen is epitope, means unless there is a complementary, it won't bind. Okay. Now have a look. Oh, more details, how it is binding. Lock and key, exact fitting, so that it won't escape once it is captured. Antibodies fit and bind like lock and key. The body does not produce antibodies that bind to its own antigens. Therefore, we only capture foreign. See, we don't capture our own cell. Of course, something goes wrong, we capture our own cell and it becomes autoimmune disease. It is a disorder, not normal. Vaccination throughout the world. We are trying to remove all the pathogens by increasing immunity. Vaccination, giving the vaccine or antigenic pathogen which is inactivated, then body system will protect, as we studied earlier, and gets rid of that little microorganism or chemicals, but keeps memory. Okay, but some pathogens which the body is not prepared can conquer and cause disease or disorder and cause serious. Polio has been reported to be caused by polio vaccine in some cases, very few cases. Vaccination means teach the system by this is foreign and you are self. Vaccination is important. First, primary. Second, prevent from getting sick. Third, life-threatening disease, measles, polio, tetanus, whooping cough can be prevented, and society at a large. Now we are waiting for the COVID vaccine. Similarly, what is a vaccine? Define. Preparation of the causative agent, especially for vaccination, to increase or bring about immunity. Now, we may have an agent which looks like disease-causing microorganism. How do we get that? We break it up, we make it weak, okay, we combine it with other chemicals. So that is how we prepare a antigenic dose. Now have a look. Vaccine, weakened form, dead or living, weak are put, body reacts, makes antibodies. Now if it comes again, our body is ready to fight with it. Have a look. Immunization, vaccine and vaccination. 3 definitions at one shot. What is the difference between vaccination and immunization? Every time we are using the word. Vaccination means giving antigenic material, and immunization giving is by giving a vaccine. You are making him immune. You develop immunity and by vaccination, and vaccination is a process of immunization. Vaccines are considered to be safe. They're thoroughly tested in our underdeveloped countries. Round. Side effects generally rare. You just get fever, pus or something like that. Nothing greater than that. But 1 in a million, they may cause reactions. Now, antigens on blood cells. Your blood cells contains certain antigens. And they give rise to blood groups. We have got A, B, O blood group. After that we have RH, Duffy group, Kidd group, Lewis group, P group, MNS group, Bombay blood group. These are minor. This is major. This will not cause much death or something like that. This causes death. That's why we study only A, B, and RH, or they are causing problems. ABO blood group was discovered by Landsteiner. They started mixing blood together and they found clotting and different clotting. So they found ABO. This gene is present on chromosome 9 for need purposes. Let's have a look at the blood group. ABO. O has no antigen, B has got this type of antigen, A has got another type of antigen, and both are present in AB type on top of the RBC. First understand the antigen. Now what happens? So when he has blood group A, he makes antibody for B. And when he has blood group B, he makes antibodies for A. See the shapes? You react to the opposite, you don't react to self. When you have AB, you don't make anything because you have both the information. And blood group O has no information, so he has got problems with both. RH factor. RH is complex. It is an antigenic protein. It was discovered by Landsteiner and Wiener in monkey, so that's why rhesus RH factor. It's also called D antigen. Those who have this factor are called RH positive. Those who do not have this factor are called RH negative. So, Levine and Stetson first talked of it, and then Landsteiner and Wiener found it in monkeys. 85% human RBCs get agglutinated, 15 do not. So that means there is some protein is different. The antibody is same in guinea pig and rabbit and monkeys. Later we know that they are not same. They are similar, they are not same. But we kept the rhesus name as it is, we did not change. Rhesus. We learn most from monkeys, so where we discovered this. But some people don't have it, so that's why you are a positive or a negative. So blood group A with rhesus, blood group A without rhesus, blood group B with rhesus, blood group B without rhesus. So plus means you have the rhesus, minus means you do not have the rhesus protein. Now see the problem. Hemolytic disease of the newborn, RH incompatibility. If the mother is RH negative and the father is RH positive, and this is dominant positive, so the child will be positive. Now with placenta, blood exchange between mother and child, but cells don't get exchanged like that. But when you are delivering, when you cut the placenta, few drop of blood will travel. So first child will be born and now mother will make antibodies to the plus because the antigen entered her system. So second child onwards, problem will start. So let us see details. First, strong immunogenic response, the baby dies. Second, important in blood transfusion also, and hemolytic disease of the newborn, HDN. Name was RBC blastosis get burst in fetus. Okay, erythroblastosis fetus. So the child cannot get born. When an Rh negative mother conceives Rh positive, if the child father is also Rh negative, we have no such problem. If the mother is positive, we have no problems. Okay. Now, as I told you, the RBCs may enter the mother system. She will produce Rh antibodies. So subsequent, first one will get born. When you cut the blood exchanges, you will get the problem, and she will not be able to carry the pregnancy. The child will be born dead in the womb. So this is what we are showing you. See, Rh negative, then antibodies form, then from this, and then the body will throw out the baby. More in details, showing you how exchange will take place on cutting. When they cross the placenta, problem starts. Remember, we are supplementing it so that you will not forget. So this is Rh in summary, so you can read all at a stretch. Let's come to disease. Disease is disturbed or deranged, totally gone or slightly disturbed. Okay, some organ or system of your body, diet, heredity or infection, we don't know, but your body is now disturbed. What are pathogens? What are vectors? From lower classes, nothing new. And what is the incubation time? What is a parasite? And two types of parasite, outside the body, inside the body. Okay, with examples given to you. Now, disease may be congenital from birth. They are permanent and practically we cannot cure them. We are trying gene therapy, enzyme replacement therapy, but these are at an experimental state. Okay. Acquired diseases you get after birth, they may be communicable or infectious or non-communicable. Okay, communicable you get directly from the infected person. Virus, bacteria, fungi, helminth worm cause it. Example we know is malaria. Okay. Non-communicable, we get it directly or indirectly, like cancer, deficiency, but we do not get it from another sufferer. So once again, for remembering, summary has been put. See, congenital, color blindness, thalassemia. Okay. Distinguish between the two. Pass from one person, cannot pass. Infectious, okay, non-communicable. They are acute, these are chronic. Lower socio-economic group, poor people. Generally, rich people will show all this because of the smaller society. Here, vectors, here dietary habits, environment, lifestyle. Examples. So this is how you write distribute between in the exam. Have a look between the two. Example shown on the left and example shown on the right. Okay, those which are communicable, those which are not communicable. So this will stay in your brains. Malaria, vector-borne. Mosquito is a vector and the agent is a protist. Plasmodium, have a look. Plasmodium falciparum, Plasmodium ovale, Plasmodium malariae, Plasmodium vivax. Both type present in the RBCs, you can see. Okay, and this is that mosquito anopheles which causes it. Incubation period is different for different. You can see the differences for your need purposes. Okay, 9 to 14, 12 to 18, 18 to 40, 11 to 20. So each one takes its own time to show the symptoms. So 4 species are there. Only falciparum is problematic. Okay, but there are 4 types. Okay. What are symptoms of malaria? Have a look. And please write some. When they tell you write 2 or 4 symptoms of malaria. Okay, spleen enlargement. The WBCs are now captured the antigen and see the spleen gets enlarged because of that. Copies of WBCs being there and trapping the pathogen. 7 to 15 days after biting, symptoms we have already seen. Last you get vomiting and you start shivering, convulsions, you get joint pain and your RBC level is low, anemia, and then liver gets enlarged because of breaking down of iron from RBC, then problem in the eye and then brain also, you start blabbering, okay, and you start reciting all your NEET and IIT preparations. So this is what happens. High fever, sweating, sudden shivering, 4 to 6 are there. You become like normal. Every 2-3 days you get whenever the toxin comes out in the blood. So cold stage, fever stage, sweating stage, and you become normal. See? So throughout the day you show, how are you? I'm feeling very cold. How are you? I'm having fever. How are you? I'm sweating. And then you become normal. Again repeat the cycle after 2-3 days. Diagram for labeling and writing in the exam. You see? So here we can see what's happening. Mosquito. Okay, here we are. So microgamete, macrogamete, fuse, zygote in the stomach lining of the mosquito, ookinete, oocyst, the cyst burst, and salivary when he bites first, the liver cells which are the main cells, hypnozoid, hepatic dormancy, mature liver, schizoid stage, merozoid, then they go to RBCs, tropanozoid stage, erythrocytic schizoid stage, and then the RBC burst, cycle continues. Okay, now have a look. First infectors, we'll start here. First mosquito bites the first person, liver cells, then RBC, then second person bites, goes to second mosquito bites, goes to second person. And this is how it goes on spreading more and more in number. So it shows here reproductive stage and the asexual stage. Reproductive stage where it is happening, and we are showing asexual stages. Transmission cycle in brief, same thing, first person, second person. Keep supplementing your information. Female Anopheles mosquito, because reproductive cycle, and it has to bite. It transfers sporozoites which multiply schizogony in liver cells and RBCs, and merozoites are formed. So the cells formed within erythrocytes which are in RBC, they behave like gametes, gametocytes, and undergo gametogony. These gametocytes, when a mosquito, another one bites, then will enter the mosquito's stomach and fertilization will take place, diploid zygote will be formed, oocyst is formed, and then sporozoites, and again the cycle repeats through a salivary gland when she bites, which we showed you in the diagram. Now, how do we know malaria? First slide we prepare of the blood and we see the parasite, and we also have now nucleic acid amplification technique to identify, and treatment, artemisinin based combination therapies. This chemical reduces the plasmodium. Combination drug cure is there. 5 different type of medicines are recommended: artesunate, sulfadoxine, pyrimethamine, etc. And old people, cinchona plant, quinine was used. Quinine has come back, okay, for COVID treatment now. Prevention, you use a net, insect repellent, spray the insecticide, drain away all stagnant water in pool, tile, pots, etc. because the larvae will develop in that of mosquito, and use gambusia fish because it eats up the mosquito larvae. Biocontrol for your need purpose also. Remember, World Malaria Day is 25th April. Second disease we see, amoebiasis. Amoebiasis is also called amoebic dysentery. It occurs in your stomach and intestine. 15% population in our country. And the amoeba is called Ant amoeba histolytica. what happens? Diarrhea, gas is passing, mucus comes out, pain in the stomach, blood comes out in the feces, and liver gets swollen, and you get fever and pain in your stomach. How do we get it? Feco-oral route. Okay, your feces get mixed and come through the food or water you ingest. Dirty hands and contaminated food. People wash there and people drink that water. That is why, okay, and where you don't clean, etc., like school taps and all that, so you also get problems like this. Pani puri wala, be careful. Okay, let's have a look how it is passing. Life cycle. Okay, so first it gives rise to trophozoite, binary fission, pre-cystic forms, cyst is formed in favorable condition, 2 nucleus seen, many nucleus like that divide, quad, 4, and then each one forms cyst. Now metacystic, lot of cysts are present, they divide into small amibulae and the cycle repeats like this. How it happens in man, we will see. First you take in by contaminated food or water, then exocystation in small intestine occurs, where the trophozoite and all what I explained happens here, and then it goes to colon, it goes into the waste matter and continues through the water cycle into food and water. First we check the stool, so we can see amoeba in it. Second, we treat with metronidazole and tinidazole tablets, Norfolk TZ as we call them, and they get destroyed. How do we prevent? Wash hands after toilet, second boiled water, third chlorinate or filter, don't eat pani puris and chutneys from the sandwich walas or making it on the road, wash and cook the vegetable, keep your toilet areas clean and dispose the sewage properly, not in water. So again, by diagram we are showing you so that you do not forget what should be done to prevent. Let's go to the third disease, ascariasis, or we see this roundworm problem, human intestine tract. Roundworm Ascaris lumbricoides is a nematoda. Male, female, see the length, see the coil, we will see there are different morphologies. Okay, that's why I put both the diagrams together for you. Symptoms. First, they attach to your walls of the large intestine. Second, they move up to the lungs. Fever, breathing problems, coughing, pneumonia. Now, third stage, they enter into small intestine and become worms and remain there nicely, subtenant. Then you get abdominal problems, intestinal blockage, partial or total also, you can't pass food through it. Too much pain, vomiting, restless, disturbed sleep, and worms come out in the feces, and when you vomit also, worms will come out. Such extreme chronic case. Okay, again put for you. When it enters the lung, pneumonitis, your alveolus walls get inflamed. Your WBC, eosinophils increase. Your hunger goes and your weight falls down. How? Food and drinks. You take the eggs will hatch and then they will enter the organ and they become part of your body. Your body does not recognize it as foreign. It's got cuticle on top, so it's unable to fight with it. Showing you the life cycle. Okay. First, fertilize the egg in the feces, larva in the soil, you eat the eggs, intestine, vein, lungs, trachea, esophagus, and then enter the intestine. Again, diagrammatic, nicely made, so that you can remember. Remember those 2-3 times we are showing you is so that you supplement. It should become by heart. So when you see, you can label the stage, what is happening at X, what is happening at Y, and you are able to draw the diagram. Remember, you don't have to draw all the pictures and all. You only need boxes with the labelings. We see under microscope, you see the eggs. And if the worm has come out, of course, there is no problem. You can see it. Treatment, piperazine, mebendazole, levamisole, Pyrantel are new tablets which are available, which you take. Now, earlier we should take 3 tablets, now it is 1 tablet, and then after 3 weeks another tablet, so that you break the life cycle. 3 months you should control it. Prevent, do not pass feces in open space. Wash hands, we keep telling you for COVID also. Wash the vegetables. Don't eat raw vegetable directly from the farm field where feces etc. or pathogens may be present. Once again, so that you can see and behave properly. Okay. We come to the next disease, filariasis or elephant foot. Okay. Let's see. First, caused by again a nematode, and the nematode is passed by Culex mosquito. Okay. So filariasis. See, we have drawn Filaria imitis and Myzomia supertica pathogen vector. 3 subtypes: lymphatic, subcutaneous, serous. Another names are given. Okay, and which species cause it? Lymphatic is the one which you call elephantiasis. Wucheria bancrofti, Brugia malayi, Brugia timori. Different country, different names have been given to the species. Have a look how it enters the. First, edema, water gathers, skin becomes thick, tissues become thick, leg, arm, breast, scrotum gets affected, all the soft connective tissue areas, and enters the lymphatic system. Lymph vessels and nodes become big, and you look like legs of elephant, and fluid causes more swelling, and in testes and scrotum and all you have hydrocele and lymph fluid. Testes appear very large. How it gets to you? Female Culex mosquito. Then they arrive on the skin. When bites goes through your skin, to molting, they become adults. They settle in incubation period. Body treats it like self, does not destroys it, and 8 to 16 months it will live in a body and make a lot of damage, which will be mostly permanent. Life cycle again explained. Stages, how through mosquito bite and blood it enters the body. We have made clearer diagram for you to understand. See, picture wise. How do we find out? First, till now, only we have to, around midnight, we have to take blood and then only we can find. Okay, because nocturnal behavior. So, we use now finger prick blood test any time of the day, new ones. And ultrasound, we can even identify the rapidly moving worms in the system. Okay. Treatment. Diethyl carbamazine citrate twice a day for 3 weeks and then 5 days every 6 months to prevent. Avoid mosquito bite is the main thing. Once you get, it's very difficult to remove it totally. Let's come to typhoid. Typhoid is infection of the intestine. It is caused by Salmonella typhi, a gram-negative bacteria. It is found in the intestinal lumen, inside the intestine of infected person. It shows O and H antigens. Okay, flagella is the H antigen and the capsule is the O antigen. Salmonella typhi is the organism. Everything marked in color and bold is for NEET and MCQ. Signs: high fever, nausea, fatigue, abdominal pain, constipation or diarrhea, rose color rash on the skin, tongue is white coated, cough, loss of appetite, breathless, irregular heartbeat, blood leaks, hemorrhage. Remember 105 and all it crosses the fever. What are the symptoms in diagram? Please have a look. All don't mean that you've got typhoid. Okay, but these are the general symptoms. See the pink color rashes. Then we come to know. How is it transmitted? Food and water. Flies and cockroaches feed on fecal matter and they transfer it to the food. Poor hygiene, poor self. Never eat open food on the road, vada pav, etc. because it is not covered. Diagnosis. Vidal's test, is a serological antigen antibody test. Okay, and it is caused by Salmonella genus, and we can see the agglutination reaction. Treatment: we have to severe cases remove the gallbladder, and chloromycetin is very useful. Okay, 2 vaccines are now prescribed by WHO: oral for typhoid 21A vaccine, and injectable typhoid polysaccharide vaccine is prescribed. How to prevent? Wash, drink, boiled, wash fruit, vegetable, and best get vaccinated. Because once you have the vaccine, this does not mutate much, it's a bacteria, and you are immune. Let's come to pneumonia. Pneumonia is inflammatory condition of the lungs. Various pathogens cause pneumonia. Pneumonia is a situation. Virus, influenza, adeno, parainfluenza, respiratory syncytial virus, all can cause pneumonia. Bacterial diseases like Streptococcus pneumoniae, fungal pathogens, pneumocystis jirovecii, and pneumocystis carinii can cause. Chemical burns or physical injury to the lungs can also lead to pneumonia. You see, the pathogens and the lung shown for you to remember which cause pneumonia. Again, various how they look like, so it's easy for you to remember, and every time you get stuck you can have a look at a diagram, so it will stay in your brain. Symptoms: greenish or yellowish sputum, high fever, dyspnea, shortness of breath, chest pain when you breathe deep, fatigue, appetite goes, headache, vomiting, joint pain, muscle aches. Have a look at the diagram which shows you all the symptoms. That easy for you to remember. Okay? Picture speaks 1,000 words. So when you watch this, and when you please take down with a notebook and a pen. Don't watch this like a movie. We are not promoting Bollywood or any other movies here. Okay, this is not Netflix, Amazon Prime, etc. This is our career and future life. Please take care. Mode of transmission. Carrier will give you, patient will give you, okay, some device which we are using you pass on, or we live together and we get co-infection. Treatment. We have to see who caused it. Bacteria, we use benzyl penicillin, ampicillin, chloramphenicol. Okay. Virus and all, difficult to treat the root cause. Prevention. Vaccination against Haemophilus influenzae, Streptococcus pneumonia in first year of life. Then you become immune to pneumonia. Remember, not burns and viral ones, only bacterial ones. Hygiene. If you do all this, you will help yourself not to cause pneumonia. Let's come to common cold. First, viral infection of the upper respiratory region, also called nasopharyngitis, acute viral rhinopharyngitis, acute coryza, or a cold. It is caused by your favorite coronavirus group, rhinovirus, coronavirus. Symptoms: cough, sore throat, running nose, fever, nasal congestion, can't breathe, sneeze, red eyes, conjunctivitis, rashes on muscle, fatigue, headache. General symptoms are plenty. Okay? Symptoms, diagram, again for you to remember. Low grade fever, 100 fever. Okay? Again, more symptoms. Remember, 2-3 times put so that these have come in the exam. So you must, they appear silly to you, then while writing, people forget how to write. Everybody will just write fever, fever, fever. That's not correct. How do you prevent common cold? Stay away, wash hand, handkerchief, and alcohol-based sanitizer. As today also for coronavirus, we are prescribing all this. Again, diagram wise, please wear a mask, wash your hands. Vaccine is going to come shortly for your COVID-19. Be strong, have vitamins, and make your immune system strong. Now we have ringworm, the troublesome character. It's a fungal infection of the skin, dermatophytosis. And the fungus are trichophyton and microsporum. They live on the keratin protein of the dead cells in the skin, hair and nails. You see? Lesion is there. And the lesion you will see microscopic under microscope ringworms. Okay? The fungus live only on the dead tissues, scab on top of the body. Okay? Red ring is caused, scaly lesion is there, itching is there, and when it is going in the nails, it will become onychomycosis. The nail will become thick, different color and shape will go. Athlete foot, because they keep wearing the socks and shoes whole day, the sweat accumulates between the digits of the finger. So you get athlete's foot, fungus between the digits, white dead tissue forming. Okay? See how the scabs look on the skin. How does it go? Sharing of clothes, comb, close contact with infected fungus, not washing, keeping the skin wet, and of course, if you try rainwater, sewage water, if you keep your feet and nails of the feet, it will also get infected. Diagnosis, physically you can see this, and we use nystatin, fluconazole and itraconazole, etc. Antifungal creams are available. Okay, again slide showing you how to prevent. Use fungicidal soap instead of ordinary soap, and boil water, wash your clothes, etc. Don't share personal items. Let's come to dengue fever now. Okay. Dengue fever is painful, breaks you down, vector-borne disease, 4 types of virus. And female Aedes mosquito. Please remember Culex, Anopheles, Aedes and the disease individually they cause. Each can't cause everything. Now again it will bite, suck blood. It cannot spread directly from one person to another person. You need the vector mosquito. You see? Mosquito bites and transfers it. Just like malaria. See this guy? If this mosquito is not there, you can never get dengue. No other way. So we come to cancer disease. Okay? Cancer, what is a tumor? Abnormal, uncontrolled and purposeless. Cells are just going on dividing for no rhyme or reason. So you get a mass, and this mass of undifferentiated cells are not doing any job, and it is called a tumor. So definition, cancer is abnormal growth of cells which affects the adjacent tissues and distant organs and finally causes death. Okay, from when it can't be taken. See how they are just multiplying and occupying space and pushing everything up. When it is malignant, it has entered the bloodstream and gone to other tissue. It is dangerous, harms the body, and it is called neoplasm, except leukemia where only the WBC is involved. Now they affect each and every organ by taking away the nutrition and occupying the space. They release secretions which change the body and helps itself. It is one of the major killer diseases today. You see normal cell versus cancer cell. See cell division occurs, we stop, they do not stop. We differentiate, they do not differentiate. Oncologists are the people who treat cancer. And tumors may develop anywhere in the body. And all tumors are not cancerous. Some are benign or non-malignant, like we've got moles, etc. on the face. And cancerous or malignant are ones which will spread and cause metastasis and death. Many slow, may become large, but remains there only, does not spread. Okay? Some may later become harmful and fatal, at least brain walas will cause death because brain gets squeezed. Okay? Sometimes they become malignant, like adenoma and fibroid, and then they change. Benign means non-progressive. Okay? It does not show the 3 properties of uninhibited division, no contact inhibition, etc. Okay, so it does not invade. So it does not metastasize. When the growth rate is very fast, goes on dividing, then mortality rate will increase. Overcrowding, you are just destroying normal space of normal cells. They compete with the other cells, take away the nutrients, and they kill them. They go from organ to organ through blood or lymph. And this is called metastasis. No treatment for metastasis. So benign versus malignant, please distinguish between. And you will have to write points like this. Of course, make a sentence. Don't, these are for remembering. 5 types of cancer. Carcinoma epithelium, breast, lung, stomach, skin cancer. Sarcoma, connective tissue, bone, osteosarcoma, muscle, myosarcoma, cartilage, chondrosarcoma, adipose, liposarcoma. Lymphoma of lymphatic tissue, lymph nodes, spleen, and tissues which are normally responsible for your immune defense. Leukemia, blood cancer, WBCs or leukocytes. Okay, the WBC count is in millions. Okay, and types: monocystic leukemia, lymphoblastic leukemia, and adenocarcinoma in the thyroid, pituitary, adrenal, and other glands, glandular tissue is C. So again, types of cancer: carcinoma, sarcoma, lymphoma, leukemia, and adenocarcinoma as an exception. Causes. Sorry, do not know much. Only we know something. Cause cancer, they are carcinogenic. Such chemicals are nicotine, caffeine, polycyclic hydrocarbons, some sex hormone steroids, given a large amount. Breast cancer is related to hormonal changes, and women who do not do breastfeeding show more breast cancer. Viruses with cancer. Now, viruses have genes called viral oncogenes, VONC, and Epstein-Barr virus, human papilloma virus, etc. are the known examples. Oncogenes are also present in your body. These are cellular oncogenes, C-onc, proto-oncogenes, which will develop into oncogenes. They are activated, and then you will get cancer. So, a mutation of a proto-oncogene causes it to become cancerous. Radiation is a major known cause. X-rays, gamma rays, cosmic rays, ultraviolet rays. Skin cancer is high in people with no melanin, white skin, and working in sunny areas because UV radiation in the sunlight causes all these moles and beauty spots to develop into skin cancer. Addiction, smoking, tobacco, alcohol, drugs, marijuana, steroids are known to cause cancer. So all causes are put for you to understand, so you can prevent and use them. So have a good diet, avoid tobacco, avoid alcohol, please go under sunlight to get vitamin D, do not go near radiation, avoid pollution, avoid viruses, like that. What is the treatment? Surgery, radiation, hormone therapy, chemotherapy, and targeted therapy. Either local or systemic. Local, very small, local, found in a particular place. Systemic, where the whole body is metastasis is being seen. So we'll see. Chemotherapy, we give anti-cancer drugs, which stop cell division. They are killing more cancer cells than less normal cells. This will cause hair loss and anemia. But once the treatment stops, you will become normal. Radiotherapy, like we use X-rays and radiations. Cobalt, iridium, iodine, radioactive is used. X-rays and gamma rays enter these cells, okay? But normal tissue gets damaged less. Of course, all get damaged. Cancer tissues are more prone and they get damaged more. Surgery, we just cut it out. Breast tumor, uterine tumor, most effective. We try our best, but once it has delocalized, traveled, surgery becomes difficult. We can't cut out all the organs. Immunotherapy, use alpha interferon, activate the body's immune system. Okay, this is also being tried. Supportive therapy, a lot of side effects are there, so we give them good food, vitamins, steroids to improve the quality of life. Okay, so therapies are required so that he can be stabilized, rehabilitated. So treatment of cancer, surgery, chemotherapy, target therapy, hormonal therapy, radiation. Remember, globally, 1 out of 6 deaths is due to cancer. Please see and then believe how bad is cancer. Okay. 9 ways to reduce your risk. Please follow these so that you have a better life away from cancer. We are coming to AIDS, autoimmune disorder syndrome. 1st December is AIDS Day. Don't forget for NEET. Fatal normally, caused by HIV, human immunodeficiency virus. It weakens your immune system, makes it life-threatening, opportunistic infections, and it is a modern pandemic worldwide. Every continent has it. Affects both poor and rich countries, developed and developing countries. Full form of it, acquired immunodeficiency syndrome. What is HIV? Human immunodeficiency virus. Full forms are required to the right for 1 mark. What is the difference between HIV and AIDS? It is a syndrome, medical condition, and this is a virus which causes the AIDS. So HIV and AIDS are not same. HIV is an agent and AIDS is a condition. It was first seen in USA, 81, and first case in 86, Tamil Nadu. So history for just your need purposes and all. What is HIV? Short note. Spherical. It has 2 single-stranded RNA molecules. It has reverse transcriptase enzyme. It is covered with 2 layers of proteins. Outer layer has got matrix protein P17. Inner layer is capsid protein. And there is additional layer of lipids there. And it has got glycoprotein GP120 and GP41 molecules. Why we are writing all